Hi class. Today we are going to talk about blood lab. I'm going to be as detailed as I can. For today, there are four main experiments that we will look at. The first one is by hemoglobinometer, meaning we will use a hemo we will use a hemoglobinometer to determine the concentration of hemoglobin in your blood. Then we will talk about hematocrit, and I'll show you on a step-by-step method how to read the hematocrit. We will also work with a hemocytometer that counts the number of blood cells. And I have made a very detailed video on blood typing, which will be posted as well. So these are the four main things we will look at. If you look at your manual on the first page of blood lab, there are terms like a complete blood count, and there are other few definitions like this. I really want you to review those few definitions on your own. Okay, moving on. So before we start, now blood lab is a critical lab because you are playing around with your own blood. Therefore, the disposal mechanisms, the disposal methods should be very good. So at the end of each experiment, I have specifically mentioned specifically mention the cleanup procedure, which tells you, okay, the blood slide goes in here, the gloves go here. Please pay attention to the cleanup methods. On your exam or on your quizzes, you might be tested on the lab cleanup part. Okay, so moving on to the first experiment, measuring hemoglobin concentration using a HemoQ. A HemoQ is this red instrument that you see here. It actually counts the hemoglobin in your blood. Okay, so hemoglobin concentration is the amount of hemoglobin in blood, and this is measured in gram per deciliter. So please highlight this and remember this. The hemoglobin concentration is always measured in gram per deciliter, and the normal hemoglobin concentration slightly varies between males and females. Males tend to have a little higher hemoglobin count, about 13 to 18 grams per deciliter, as compared to females who are 12 to 16 grams per deciliter. The reason being, we females, we menstruate every month and we lose a little bit of hemoglobin there. I have also posted links to two YouTube videos on the top. They are both very good. I highly recommend, and they are really short videos. I highly recommend that you watch these videos to get the feeling of how this experiment is done in reality. Okay, so once you are done with your experiment, you will dispose of the test strips and lancets. I have not shown you the lancet for this experiment, but for the hematocrit experiment, I'll show you the lancet. And you will see how, now there are different types of lancets available on the market, but the ones that we use at Santa Ana, you will see that on one of the PowerPoints. Okay. So the lancets and the test strips go in the biohazard sharp wastes. Alcohol swabs, band-aids, and other materials that touched blood should go in the biohazard soft waste. Please know where things go. And then other clean waste should go in the regular trash. Okay, moving on to hematocrit. Really important concept. Hematocrit is also called packed cell volume. Packed cell volume, popularly known as PCV. Now, let's say I am working in a lab and I draw blood from a patient. And let's say this is 10 mils. So I take 10 mils of blood sample from a patient. Okay, all blood. Now what I do is, now I'm not showing you hematocrit here. I'm just showing you the composition of blood. Okay, so we are talking about composition of blood. Okay, now if you take this tube and spin it down in a centrifuge, you will see that the blood separates into layers. So at the very bottom, you will have all the erythrocytes or red blood cells clumping together. In the middle, you will have a small white area, and then you will have clear plasma on top. So this is plasma. This middle part is called buffy coat, and the bottom part will be erythrocytes. Okay. Erythrocytes are nothing but red blood cells. We know this. The buffy coat will be made up of two components. Within the buffy coat, you will find white blood cells and platelets. Do you remember platelets? They help in clot formation. And on top, you will have a clear straw-colored fluid called plasma. So if you spin down human blood or blood from any animals, any vertebrate animals, the blood will separate out into these layers. You will have the heavier red blood cells at the bottom, in the middle a buffy coat made up of WBCs and platelets, and you will have straw-colored clear plasma on top. Okay, now moving on to hematocrit. What is actually hematocrit? Hematocrit is a proportion of the blood that consists of packed red blood cells. So right here, if you look at the percentage of the red blood cell, this is hematocrit. Okay, and this is always used as a percent by volume. So here we can say that 44% to 45% of the blood volume is our hematocrit value. So we will say that the hematocrit value here is 45%. Does that make sense? We will say 45% of the blood sample, hematocrit is 45%. Okay. Now, as we see here, the red cells are packed together by centrifugation. And just giving you an example, a hematocrit of 25% means that there are 25 mL of red blood cells in 100 mL of blood. So let's look at the picture on the right hand side. So we do see a blood sample which has been centrifuged, and at the bottom we see hematocrit, which is the percent of red blood cells. We do see a buffy coat in the middle, and then we have clear white plasma with water, proteins, nutrients, hormones, etc. floating on the top. Now, the value of hematocrit varies slightly between males and females. Slightly higher in males, slightly lower in females. You do not have to memorize these numbers, no. Now, if you have lower hematocrit value, as we see in the middle panel, that means obviously you have low red blood cell count, and that tells us that the patient has anemia. Now, some patients might have higher hematocrit value, meaning they have more red blood cells, and that represents a condition called polycythemia, where you have elevated hematocrit percentage. Now, the question is, Rupa, we understand this, but do we just take blood in a test tube and spin it down to read the hematocrit value? No, we do it in a different way. We use a tiny blood capillary to do this, and let's see how we actually do the experiment. Okay, so I have just put down the step-by-step method how we do the experiment. My step 1 is different from step 1 of your manual, because I have kind of deleted some steps from the manual. I have just focused on the important ones. So if your manual might say there are 12 steps for this experiment, but my PowerPoint might say there are 8 steps for a particular experiment, but that's fine. I have just highlighted the important steps here. Okay, Step 1. You take an alcohol swab and clean off a little bit area of your finger that you will poke. Step 2. You take a hematocrit tube and identify the side of the hematocrit tube that has red ring around it. This side is the clean side of the hematocrit tube, and this side will go into the clay sealant. Blood can only touch the side without the red ring. Let's take a look. Okay, so this is the tube I was talking about. Do you see these tiny, tiny capillary tubes packed in a plastic container? These are the ones we use for lab. Okay, now each tube has two sides, right? The side with the red ring, we do not draw blood with that side. We draw blood with the opposite side. So blood comes up this way. The side with the red ring will be used to be sealed. So we will seal this side with clay. Make sense? Okay. So we have a clean side. The sealed side or the clay side will be the clean side, and this will go into the clean sealant as I mentioned. And blood can only touch the side without the red ring. Now, then let's take a look at the lancet. Do you see the orange things on the right hand side? These are called lancets. Lancet. And this, we will be using these to poke ourselves. So there are again different types of lancets. If you Google, there are blue ones, white ones, red ones, but these are the ones, the orange ones are the ones we use for our lab purpose. So I just Googled a picture and posted it here. And after you are done, obviously the lancets are touching your blood. So this will go into, and it is a sharp object, right? It's poking you. So it'll go into the sharp waist. Don't forget. Okay. So with the help of a lancet, you lance your finger or you poke your finger, and then just let go the first drop of blood. As the second drop comes out, you take the tube, like the picture on the right hand side. You take the capillary tube and hold it near the blood droplet, and the blood will flow into the capillary tube by capillary action. Now remember that this capillary tube is heparinized, meaning the blood will not clot when in the capillary. And then after you fill about 3/4 of this capillary with blood, you put the side with the red ring into the sealant and you seal that side. Okay, so blood cannot run out of the hematocrit if sealed properly. Make sense? All right. Then you take the capillary tube and put it in a centrifuge, and this is a centrifuge. The picture is showing you a centrifuge which is specific for the blood capillaries. So in each one, students usually put their own capillary tube with the sealant side outward. So this part is sealed, right, with the sealed side outward, so blood cannot escape. And then you cover the lid and centrifuge on high for about 5 to 6 minutes. And after that, what do you see? You see a capillary like this. So what do you see here? You see that red blood cells have taken up the red blood cells are clumped at the bottom, and then on top you have the clear straw colored plasma. The most important thing of this experiment is to read the hematocrit using a chart. I have posted a link here to the YouTube video. I recommend that you watch the YouTube video and see how to read it. But the idea is you will align the bottom of the red with 0 and then the top of the plasma with 100. And if you trace the line where the red blood cells are, this tells us that this is 45%. So that's how we read it. I'll start with a new video and talk a little bit